So one of the astonishing facts from the Human Genome Project was that single nucleotide variants occur approximately 1 every 300 nucleotides. So if you do the math, that means there are roughly 10 million single nucleotide variants in the human genome. And these variations are one contributing factor to the genetic diversity in humans. And so this opened up a whole new set of questions. So what is the intersection between disease and genetic variants? What's the relationship between drug metabolism and genetic variation? Is there a cancer genome? And does that cancer genome change over time and in between the primary tumor and a metastasis or a recurrence? How does the genome impact mental health, behavioral health? It had already been observed that some diseases were more prevalent in certain populations. And so then questions about what's the role of genetic variation in ethnicity. And so over the years that followed the completion of the Human Genome Reference Sequence, it became more and more clear that we knew too little. We realized that there was a need for a more concerted effort to understand genomic variation. So the NIH announced its support of precision medicine in 2015 when then President Obama, in his State of the Union address, described precision medicine as an emerging approach to disease prevention and treatment that takes into account people's individual variations in genes, environment, and lifestyle. So this was accompanied by a publication in the New England Journal of Medicine written by then director of the NIH, Francis Collins, and past director of the National Cancer Institute, Harold Varmus. This perspective further supported the convictions of President Obama that science offers great potential for improving human health. And this officially launched the NIH Precision Medicine Initiative in 2015.
One of the common visions for all type of labs is that they will make use of the automation and digitalization. So the digital infrastructure needs to take into account the large volume and variety of data that is being produced. Some later on challenges can be avoided if the organizations have data centricity in mind when implementing the new systems and workflows and automations. And one of the general challenges that all labs are facing is how to capture this large amount of data that is being produced, including the contextual data, in a fair format, which means to be findable, to be accessible, to be interoperable, and reusable. And how to redesign the current paper-based workflow.
What is the future for our species? You know, in Fathi Korea's previous talk, he was talking about the factors that are involved in space exploration and how we can study the expression of genes and things like that in space exploration, but we've only really just begun human space exploration. We don't really know what happens to our species with another century or a millennium of space exploration, where our species goes. A big question is, will we see this idea of the singularity of all of us kind of merging into our machines when our technology advances so fast that we basically become the machine, or the machine takes over and leaves us behind? We don't know what that looks like. It might be likely that a lot of life out there in the universe is post-biological life, that most life is artificial intelligence, and basically written in silicon and other materials of metal, rather than being built in organic chemistry as we are. It's really important in your life and your career, no matter which field you pursue, is that you find the right mentors. And I've seen this a lot, I think, because of how we structured academia. A lot of young people, a lot of early career researchers, they think a mentor is just a teacher or an advisor, like an academic advisor, and that's really not the case. A good mentor is far more than that. A good mentor is someone who knows when to be your friend and when to be your boss. It's someone who knows how to teach you, but how to help you to learn how to teach yourself. A good mentor is someone in your life who is really going to be there for you to help you answer questions, to steer you in the right direction when you feel like you don't know which way to go, but also to allow you to grow in your own ways. And so a mentor is someone whose hindsight can become your foresight. For those of us out here in the world who've been very fortunate and blessed and honored to be mentors for young students. And
what is bioinformatics in the time of high-throughput biology? Now, modern medicine and biology has gone a tremendous improvement and evolution. I would say it's a revolution. In current age, where we have so much of high-throughput, technologies, where we can measure different modalities from, we can do imaging, we can understand tissue cellular composition, we can do genotyping. We can actually also leverage the powerful omics like genomics, transcriptomics, proteomics, not only from organs, but our tissues or cells, but also in vitro settings or in vivo settings. All these taken together, when we sequence them, we can actually build up a map or network at a cellular level, right? And these, when put into sequencing machines, can actually build a huge information complex of multi-omics information that can help us to tease out the different molecular features, which, when integrated properly, can help us to understand the disease progression.
We at Tauber Bioinformatics Research Center are a team of biologists, bioinformaticians, mathematicians, and computer scientists. And we developed this T-Bioinfo platform where we implement open source and sometimes our own algorithms. Today, we want to share with you some of these new sections that we have been working on. Our manifold utilities section. Then we will move to the single-cell RNA, jump right to spatial single-cell analysis, and then we will kind of move gears and we will speak about metagenomics analysis. And finally, talk about the importance of containers in bioinformatics.
I'd like to show you Tauber Bioinfo web platform start page. Firstly, we need to go to data integration and manifold and modeling zone when we choose the manifold utilities section. Here we can select the type of dataset. It can be object features table or distance matrix for graph, which will be used as pre-computed graph for manifold method. Let's get object features table for mice expression dataset and upload it to the platform. Let's check a visualization. Great, UMAP approach works. We can see dividing of samples into two clusters, control samples and trisomy samples.
Well, it was a great breakthrough when single cell was developed compared to bulk DNA. It allowed us to specifically study separate cells. But when we analyze data using single cell, we end up with something like this, cells in an artificial dimensions. In this case, it's UMAP, but we never really know where exactly in our tissue these cells are located. And with spatial transcriptomics, finally, we have this opportunity to map the cells to our tissue. But other than organs, we also can look at, for example, biofilm populations. This is the clustering of different functions that cells, individual cells, it's used a lot in medicine. Also, it's very useful to identify the invasive margin of the tumor and actually how the cells in this cancer interact.
So microbial tracking 2 was an interesting study, and actually that was the first human-related space biology study that was sponsored by the Space Biology Program. And so the study was looking at, as I mentioned, taking samples from the environment. So we were swabbing different surfaces, dining table, the toilet, exercise equipment, and so forth. And then we were also taking samples from the crew. So we were taking samples from their mouth, from the nose, and from different skin areas, so like forearms, and the navel, and behind the ears, and so forth. So obviously these days, so after collecting samples from the station and then those are returned to the ground, and then obviously we use high-throughput methods to be able to characterize the microbiome environment. And so here's a snapshots looking at the relative abundance, so the top 12 genera.
CHIME is a tool that analyzes metagenomics data, the culture-independent microbial communities. What we basically learn about them is composition and function of these communities. And it can be applied in a bunch of areas like agriculture, healthcare, where we can study microbiomes of different organs, For ecology, where biosensors are developed using metagenomics, and biodegradation of plastic is also a bacteria-driven process. And of course, biotechnology. So on the platform, we present 3 modules: pre-processing, that consists of filtering and denoising algorithms. Then we go on to taxonomy with silver classification, filtering our classified sequences, and conducting phylogenetic trees. And then we can move on to metrics to calculate for alpha and beta diversity.